Hello, everybody. This is Dr. Kevin Connors, and we are going to talk about the detoxification pathway genes. These are super important, especially for those who have had a cancer diagnosis, because what is cancer? Cancer is something getting inside the cell and affecting the nucleus and disrupting the replication pathway, and now the cell is in rapid replication. That's what, by the very definition, is what cancer is. So when we talk about causes for cancer, we really have to look at detoxification pathways, because the inability to get rid of things that we're exposed to, whether we breathe them in, whether we inject them in our body, whether we consume them, and it goes across our gut border, things that pass across any one of our borders that have to be gotten rid of. So they have to be detoxified. So if things aren't being detoxified either intracellularly, through my kidneys, through my skin, through my liver mainly, then they are becoming a part of us, and they tend to settle inside cells, and then they'll just sit there, and you can live to 120 and have no problem. But if it causes dysfunction to the replication cycle, then you're going to have a rapid replication, and that's what cancer is. But it could cause other things. It could cause damage to the cell membranes. It can cause damage to the receptors, and it's basically every single disease known to man has to do with cellular dysfunction. So if it's causing any sort of cellular interruption, then we need to address it. So from a genetic standpoint, it behooves us to look at that, because if I have defects in my genetics on the enzyme or the genes that create enzymes that help with detox pathways, then I'm going to have already a hindrance of function there. So that's why we need to look at it. So let's start by looking at the glutathione pathway. So glutathione is one of your main intracellular detoxification pathways, and I thank Ben Lynch and Seeking Health for this chart. So this is called the transsulfuration of the CBS pathway. It is essential for detoxifying sulfites and sulfate groups, and ammonia and glutamates, but we're not going to talk about that here. The CBS pathway also feeds down this way to create glutathione, and glutathione is then recycled, and it has to do with all these little bubbles. These are little abbreviated names for different genes. So glutathione then is released through a pathway that's not on here, called your KEAP1 and your NRF2 pathway. A lot of information about these pathways. You've probably heard of NRF2, you've probably heard of glutathione, you've probably heard of superoxide dismutase, if you've been doing any research on detoxification and such. So your KEAP1, your NRF2, your glutathione, your superoxide dismutase, and your catalase pathways are the main ones we're going to talk about that relate to this chart. Then we're going to look at some other ones as well. So we make glutathione, we recycle it, we hold it in place with the NRF2 and the KEAP1 pathway, and then along with catalase and superoxide dismutase is how we get rid of intracellular toxins, mainly superoxide ions and peroxynitrate ions. These are free radicals. So free radicals can damage our cell membranes. Free radicals can cause cancer. There's more and more research on peroxynitrate and heart disease and cancer, as well as new research that's coming out for Alzheimer's and dementia with peroxynitrate. So these pathways are very important even if we don't have cancer. So we want to look at them. So let's look at these from a genetic standpoint. So this person, our sample patient, doesn't really have a lot of defects on our initial piece on how we make glutathione. So the CTH, all these genes that we're going to look at here have to do with this pathway right here. So let's look at that. So yes, they have a few defects in there, maybe some hindrance, possibly nothing there. That's real good. Some issues here, some issues here, but overall, I've seen a lot worse. So comparing it to a lot of people in my patient base, which are mainly cancer patients, this is actually quite good. So not a huge hindrance here. Now we got to look at glutathione is being made, but is it being released in order to work to get rid of this stuff? So that is the NRF2 and the KEAP1 genes. So we scroll down some more, and let's skip that piece right there for a second. We'll come back to that. Here's the NRF2 and the KEAP1 genes. And here's where the person has a lot of issues. These KEAP1 genes may be arguably some of the most important in this whole pathway here. And this person has defects on all three of those genes right there. The NRF2, they got defects on what, 5 of the 7 there. So definitely some issues that we may want to support KEAP1 and NRF2 for the release of glutathione. So I would definitely, I checked those 2 products that we may use for this. Remember, we're not treating the SNPs, but we do need to support the function of that pathway. Scroll back up here a little bit right here, because this is where we see a major problem with this individual. This is their SOD genes, 3 genes that are the most important, and they have defects on all 3 of those genes. That's this one right here. So SOD stands for superoxide dismutase, and it grabs onto superoxide ion and prevents it from combining with uncoupled nitric oxide to make peroxynitrate. And this is the big bad guy that can cause all sorts of problems. So we need to get rid of this. This is made by our body, but it's supposed to have a very short half-life, and our body is supposed to gobble it up with the enzyme superoxide dismutase made by this gene right here. But when we have a genetic picture that looks like this, where they got defects on all 3 of those genes, they're going to make maybe a little bit less SOD, maybe produce a lot more peroxynitrate, maybe increase one's risk for all sorts of diseases. So we want to support this. Superoxide dismutase breaks that down into hydrogen peroxide, which if glutathione is released adequately, which we saw this person had KEAP1 defects, so we want to support that. And if they don't have any catalase defects, that's where it breaks down into water and we pee it out into the toilet. However, this person has a number of catalase defects too, so something else we may want to support. The neat thing about this software, again, is when we could look at, oh, well, it's 49% of the people have that defect, and 49% of the people have that defect, and 42% of the people have that defect. But how about overall? We can click on this little button and see where they rank as far as the mean of the population. And again, this is 25,000 people in this software are not a healthy cohort study. These are people that are ill that are running this test. So really, healthy mean would be somewhere over here. We never really want to be to the right of this mean. This person is fairly far to the right of this mean. You know, I've seen people with 222 here, so it can get worse. But having defects on all of those is definitely going to be a significant piece to this person's puzzle, at least that we want to look at. So that's the gene defects that can hinder the detoxification of free radicals, reactive oxygen species, peroxynitrate, and those are very cellular damaging products. Now you get into mainly liver cytochrome P450 pathway defects, and this doesn't look quite so bad for a person. And this ABP1 gene defect, I talk a lot about this in other videos about how this is your main pathway for getting rid of non-steroidal anti-inflammatory drugs and some other drugs. People that have a 2 on that pathway, I really have a long chat about. Not taking any Tylenol and Advil and things like that, because those are the people that can have acute liver failure because of that. This person wouldn't have to worry so much about that. The PON1 pathway is a key pathway for me when I look at cancer patients, because this is how you get rid of, I know the software here says pesticides, but it's really large chemicals. Pesticides, insecticides, and they're everywhere. Even if you're trying to eat a completely organic diet, you're still exposed to those things. But this is the pathway that you get rid of vaccine ingredients, different medications, large chemicals that you're exposed to. So Very important pathway. Matter of fact, I put on the website a video just on the PON1 pathway and how important it is, not only in the liver, but through the bloodstream, intracellularly and intercellularly. The PON1 pathway, the gene that's made through this pathway, is super important with cancer. The NAT pathway 1 and 2 are also important in detoxifying medications. So this is a very important pathway for cancer patients as well. Remember, anything that gets inside the cell that disrupts replication cycles are at cause for cancer. So we want to get rid of things that we're exposed to. Yes, we want to decrease our exposure. That has to do with lifestyle changes, but we want to help improve these pathways that can cause problems. And we have to talk briefly about the MTHFR gene because this is another piece here of detoxification, and this person has a 2 on one of the 2 MTHFR genes. So definitely this person, we want to support some liver detox pathways and cellular detox pathways. Products that we choose depend upon testing and other variables that we look at, as well as the person's diet. So as a holistic approach as we possibly can take. Make sure you watch the other videos in this series too, and we will talk again very soon.